Hello, everybody, and welcome to the Replace Educational Webinar Series. So my name is Mieke van Milders. I work at Cienzano, and I'm here today to give you presentations and the introduction to the speakers of our webinar series. So while we are still waiting for some people to join and to enter the webinar, I would like to give you a bit more background information on what is Replace. So Replace is a project coordinated by Cienzano and the Vrije Universiteit Brussels, which aims to collect all available expertise on the use of alternative methods to animal testing, also known as NAMs or New Approach Methodologies, in one central database. So this includes different types of technologies, such as in vitro methods, in chemical assays, in silico modeling, AI, but also other emerging techniques such as high throughput testing strategies, omics, and so on. So one of the key pillars of our project is to increase knowledge sharing and promote the use and development of NAMs. And for this, we are organizing many different types of activities. And one of these is then the Replace Educational webinar series. So this is actually the second session of this year. And in the fall, we will continue with 3 or 4 more sessions. If you're interested to be updated about our ongoing activities, you can go to our website and register to our newsletter. So before we start, I also have some housekeeping rules. So during the webinar, there are always 2 speakers. So every speaker will have 10 minutes 20 minutes for the presentation and 10 minutes for questions. So you can always put your questions in the chat because all participants are automatically muted. All the webinars are automatically recorded and then they are placed on our YouTube channel, so you can also watch them afterwards. The webinars are also accredited by the Flemish and Brussels region in Belgium. So if you would like to have a certificate of attendance, don't be shy to let us know, and you will receive it within 2 weeks after the webinar. So if you don't receive it the first few days, you don't have to contact us. It will be at latest within 2 weeks, maybe a bit longer with the bank holidays coming up next week. But don't hesitate to contact us in case you didn't receive it after 2 weeks. So now we can start with the first speaker, which is Professor Steven van Cruchten. So Steven is a professor at the University of Antwerp with prior expertise as a reproductive toxicologist at Janssen Pharmaceuticals and AstraZeneca. His research focuses on developing and validating translational models, including NAMs, for drug safety assessment in pregnant women and the pediatric population. He is also currently the president of BELTOX, so the Belgian Society of Toxicology and Ecotoxicology. And he actively contributes to progress in the field of regulatory toxicology and drug development. Steven, the floor is yours.
Thank you, Mieke. So I will try to share my presentation. So normally you should be able to see it in...
Yes, we see it.
Perfect. So the focus of my talk today will be on developmental toxicity testing and NAMs that are used for this purpose. I will discuss the assay that we've developed in our lab, but I will also put it a bit broader in perspective and show you what are the other ongoing efforts regarding NAMs and developmental toxicity testing. So if we talk about these tests for developmental toxicity, so the aim is, of course, that we will prevent birth defects that are caused by xenobiotics, so by pharmaceuticals and by chemicals. And I think I just included here 2 examples. The left one will be very familiar for you. So this is a baby that was born when the mother was taken in thalidomide, so Softenon, in the first trimester against morning sickness. And then on the right side, you see another example of an effect during the first trimester when the mother was taking in isotretinoin. So that's actually a drug against severe acne. So for thalidomide, as you probably know, the characteristic is, I don't know if you can see it, I will put my pointer on. So typically is that we have phocomelia, so it means that we have these underdeveloped limbs and mainly the arms. And for isotretinoin, it's microtia, which is very characteristic, but also, as you can see, parts of the skull and the face are abnormal, and that's because of the interference with the migration of the neural crest cells. So in the end, we want to have actually model, and I call it then the Holy Grail. So a model or a combination of models that will actually prevent or predict what will happen in a pregnant woman. And for chemicals, this is really crucial because, of course, we do not expose women deliberately to chemicals. For pharmaceuticals, you could think, well, women will also take it then for certain diseases. But we know that at this point, pregnant women are often deprived from the pharmaceuticals just from a safety point of view. So we, in most cases, do not have the clinical data, and we need to rely then on models that will predict the potential effect. The gold standard at this point in time is still animal in vivo studies, and in most cases you need to perform a study in pregnant rats and in pregnant rabbits. And this has also been taken up into the regulatory guidelines and as such reflected by them. I won't go into detail regarding the guidelines, but I want to show you just 2 slides to be aware of what is actually done in the in vivo studies in order to understand what you need to mimic then also with the new approach methodologies. And I've just included here one guideline, so that's the ICHS-5, so that's the one for pharmaceuticals. I could also have shown you the one for chemicals. The principle is actually very similar. When we talk about pharmaceuticals, we talk about an embryo-fetal development study. When we talk about chemicals, it's a prenatal development study. And the exposure period is approximately the same. So for the pharmaceuticals, it's during the period of organogenesis. And for the chemicals, you will extend it until the end of gestation. I've included this just to show you that you do not only look at malformations in this study, but also at potential maternal effects in these EFD studies in order to be able to potentially discern effects which are driven by the mother or effects driven by the mother and others directly driven by the compound. And this is also shown in this very busy slide, and I won't go through all of the endpoints. But as you can see here below, that's the 3 below parameters or endpoints are the fetal evaluations. But you will also look at fetal body weight, so the fetal weight. You will also look at the placenta. Eventually, if you have any resorptions or life or death conceptuses. So it's much more than just looking at malformations. In 2020, the guidelines, so the ICHS-5 has been revised in order to also provide opportunities to use NAMs for regulatory submissions. And again, you see here Annex 2 depicting the potential scenarios. And you can see that it's only under limited circumstances that you can actually use those for regulatory purposes. Still, I mean, there are some interesting cases depicted here, and I will just show you 2. So imagine that you want to perform your animal study, but actually you cannot reach the exposure that you intend to have actually in humans, so that you are below the therapeutic concentration. Then, of course, you have a problem in view of risk assessment, and then potentially you could use a NAM at a higher concentration in order to provide some data on safety. And also this one is quite interesting. So imagine that you perform your animal studies and your findings are equivocal. So it means that you cannot make any strong conclusions. Then you could actually add maybe a NAM or more NAMs actually to increase the weight of evidence and to decide whether there's a problem, whether there's a risk for the pregnant woman or there is no risk for the pregnant woman. So these scenarios are quite interesting, but at this point in time, we see that companies are not really using those scenarios. And that's a bit strange in the sense that, so you've seen that this guideline has been updated in 2020, but when we talk about NAMs for developmental toxicity, already 3 of them have been validated in 2000 for developmental toxicity. So the mouse embryonic stem cell test, the MEST. Then in the middle you see the RWEC, so that's the rat whole embryo culture. And here on the right side you see the limp butt micromass test. And to be honest, the right one is less or has less been used. But the mouse embryonic stem cell test is still used by companies for screening purposes, and in some cases also the rat whole embryo culture. I won't go into detail regarding the setup of these different NAMs, but just briefly for the mouse embryonic stem cell test, you will isolate the inner cell mass, so the embryonic stem cells, and then you will expose them to the compounds, and you will see in the end whether, in most cases we look at cardiomyocyte differentiation, where these cardiomyocytes are contracting. So that's like a very rough endpoint which is assessed. And the advantage is, of course, that you only need a small amount of test material and you follow the principle of the 3 Rs. The disadvantage, of course, in this particular case, as I told you, cardiomyocytes, it's only one cell type, so it's not the whole organism that you will actually evaluate. For the whole embryo culture, the advantage is that you have complete embryos, so it's a whole organism. But the disadvantage is that you will only expose those embryos for a short time, so for maximum 48 hours, and that's not covering the entire organogenesis period neither. And you use, of course, pregnant dams, so pregnant rats, to isolate the embryos from the womb. And then the last one, so the micromass limbed assay. There also you need just a small amount of test material, but you will so isolate the limbs from the embryo, so it's also not a whole organism, and it's also not a replacement because you are using still pregnant dams for this purpose. So the question then arose whether there is any alternative method that could actually be a whole organism and where you don't need to sacrifice the animals, and that's where the zebrafish embryo actually comes in. So when you think about the zebrafish embryo, so of course it's a fish, but it's still a vertebrate. So that's the same as for the mammals. It shows a vertebrate body plan. It has a very small body size, which makes it also easy to handle these fish. And on top of that, we have several other advantages. So because they have been used before for a lot of genetic disorders, the genome is fully sequenced. We have a high reproductive rate, so it means that they can produce one pair of fish, a male and a female, easily 200 embryos at once. And then a major advantage is that you can actually follow up these embryos over time just through the stereo microscope. So you can actually do a time course experiment where you can follow them up. One of the major advantages that has driven to use this model is actually that it's considered to be not to be an animal until 5 days post-fertilization according to the European directive. So it means that if you stay within this time frame, so beyond not beyond 5 days post-fertilization, it's not an animal experiment, and it's open for medium throughput, and it has a very fast embryonic development. So actually organogenesis in this model is almost completed by 5 days of fertilization. So there are some organs which take a bit longer, but you cover almost the entire organogenesis. So very useful. Within our lab, we also developed this assay. So it's called then the zebrafish embryo developmental toxicity assay. And you can actually assess several malformations. You can see them listed here. I won't go into detail. I just included a picture below. So this is a normal larvae on top, and this is a malformed one. And as you probably see, with this one, the tail is kinked, and that's something that we see for several teratogens with zebrafish embryos, that the tail is then kinked, so it has an impact on the tail. Here we see also a malformed mouth. We have edema of the pericard. You can see this is the yolk sac, and this is also the yolk sac, so it's much bigger. Edema of the yolk sac, and even here we have a bleeding, which is present. So embryo with a lot of malformations. Depending on their age, so normally you start at 5 hours post-fertilization. That's the start of organogenesis, up to 120 hours post-fertilization. So depending on the stage, you can assess one or more of these parameters, and the older they get, the more that you can actually evaluate over time. The model, of course, also has some limitations, and that's important to mention. I will come back to that. All of the NAMs have their limitations, and actually also the animals have their limitations. So first of all, there are some differences in organs. So the zebrafish has a swim bladder. We don't have this, so that's just one of them. Maybe one of the most important things is the exposure method. So these embryos are kept in a well and then exposed, so in the medium, to the drug or the chemical. When you think about ourselves or the animals, then in most cases we get an oral dosing, so it's an oral uptake, so the different exposure is definitely present. And then something that we focused upon in our group was actually looking at metabolism and pharmacokinetics. Maybe I should briefly explain this. So for the zebrafish embryo, as we have an external fertilization and we have an external development, it means that the mother is not involved at all in this particular organism. When we compare it to mammals and you dose the pregnant animals, it means that the mother will metabolize the compound and then the parent compound and the metabolites will end up in most cases to the fetus. Here it means that the embryo will have to do the metabolism itself because there's no mother that will do it. And then the question arises whether these embryos and larvae are able to do that. And I don't have time to go into detail, I will keep it short. We did a lot of work on this and we show that actually only from 72 hours post-fertilization onwards, so actually a major part of organogenesis is deprived from metabolic capacity, so that's something that you need to take into account. We also noticed that when we looked into literature to other groups evaluating the zebrafish embryo, that for the false negative results, some of the compounds were actually skeletal teratogens that we missed in the assay. So we were wondering whether we could maybe add a skeletal staining to our assay to improve the sensitivity. And then the last one, each lab has its own protocol. So there's really a lack of standardization of the protocols. And we also noticed when we went through the different papers to look at similar compounds that were tested, that actually the discrepancy between the different labs is quite high, probably due to the difference in protocols. So that's definitely a need to standardize these protocols. We did that in our lab. So again, I will try to keep it simple. So in the middle panel, you can see our standard assay. So what we actually do is we use a 48 well plate. We put the embryos at the start of organogenesis into the medium with a certain test concentration of the compound. And then we will start evaluating them over time. So here you see 5, 10, 24, up to 120 hours through the stereo microscope, and we will score them for malformations. As I told you, in these early time points, the embryos don't have the metabolic capacity. So to accommodate this, we developed an add-on system to have still metabolite production. And this was done by a pre-incubation system where we used human liver microsomes. They are then actually brought into contact with the compound for 1 hour. They are then ultracentrifuged, so it means by 100,000 G. We take the supernatant of this, and then we had to dilute it 1 out of 10 because you see toxicity of the human liver microsomes on the embryos when you co-incubate them, and even when you don't, and you don't dilute the supernatant, then you still have toxicity which is induced by the components of the medium of the human liver microsome. So that's something that you definitely need to do. For the skeletal staining, we also have an add-on system, and that's why it's called then the SZTA, and then the metabolic one, the MZTA. Here we developed a method to stain them with alizarin red, and at this point, when we published the paper that you see on top, this worked out fine for positive control, so we could actually stain them. But then I come maybe to a bit of the bad news, in the sense that we did a further validation of the ZTA, and as a consortium effort with several companies, we used several compounds to evaluate them with our staining method. And we actually had to come to the conclusion that we had a too high biological variability in skeletal development in these embryos and larvae. So that actually means that due to this variation in your normal development, you need to have a very strong skeletal teratogen to be able to depict or to detect actually the effect. And in our case, for the ones that we were using, we could not detect anything. So I think that's an important message to bring here, that skeletal staining methods are definitely possible in the zebrafish embryo and larvae, but to make it then, or to use them for toxicity assessment, this will not be possible. Other groups also have validated the assay further, even with the compounds that are mentioned in the ICHS5 guideline. And also here I will keep it short. The original sensitivity of the assay, so that means that the real teratogens were detected as a teratogen, was 79%, and then by tweaking a bit the protocol and also the time of assessment, they could increase it to maximum 88%. So the question then arises, is this like good or is this bad? And then this is a very interesting paper. It's a paper from 2018, and there they compared actually the different NAMs for developmental toxicity, so based on literature data. And as you can see here, of course, this is based on just single papers. There are several papers available, but we always like in this framework and a maximum accuracy of 80 to 85%. So the zebrafish is actually doing the same regarding the accuracy as the other ones, which are also available. So the question arises then, is our zebrafish embryo the holy grail? I think we can definitely say no, but there is a but. So the zebrafish embryo is not doing worse or is not performing worse than the other DevTox NAMs. And I think most people now within the field are agreeing that this 80-85% is the best that we can get. And the reason for that is probably because we have a caveat. So the aim of these NAMs is to predict human risk. But what we do, because we don't have the human data, is we compare our NAMs data with the animal in vivo data. And the question is then whether these rat and rabbit in vivo data are actually completely representing the human situation. So that's very important to bear in mind, that you benchmark them against actually not the right species. Then you can question, well, but does it work then, this two species approach? Well, it has definitely worked. So we can say that by using pregnant rats and pregnant dams, this has prevented teratogens to be marketed. And if they have been marketed, we had a signal in one or both of the species. But single species by itself, so just the rat and just the rabbit, is insufficient to detect all of the teratogens. So if you would take them just by themselves, then probably they also just end up with an 80 to 85% of accuracy just by themselves. And even more important to bear in mind, and that's something that's been neglected for quite a while, is that probably these animal studies also show quite a lot of false positive results due to the interspecies differences. And you can wonder, maybe for chemicals, if that's not that critical in the sense that at least you will detect the teratogens, and if they are non-teratogen, well, then you will just throw the chemical in the bin. But when you think about drugs, this is becoming really an issue, because at this point, pregnant women are deprived from several drugs because they are thought to be teratogens. And if they are not, then it means actually that these women do not get the medication that they should actually get. So because of all these things, in the most recent years, people have been thinking, well, why are we using actually animal NAMs? Why aren't we using human NAMs to predict the human outcome? So we know now that academic labs, but also companies, so here, for example, for the human ESC Stamina, and then here for the human iPSCs, Toxis have developed human developmental toxicity assays. And then also here you can wonder whether these will then predict the human outcome. What we know for the one from Stamina is that they reported an accuracy of 77%. And for the Repro tracker, an accuracy of 91% has been reported. Now, with these percentages, please be careful, because it's not the same set of compounds that they both tested. And again, this is benchmarked against the animal data. So it's not benchmarked against the human data. So what can we then actually do? That's the question, I think, when you see all these data. From a practical point of view, we see that several companies are using several NAMs for internal decision making. So not just the zebrafish, but they will combine it, for example, with the mouse embryonic stem cell test or with a human iPSC model in order to hopefully come to a better risk assessment, mainly for screening purposes. So not really for submissions, but really during drug discovery or for chemical development. And having just included one example, I hope I still have some time, to show you how this has been done. So it's a case study for an anti-malarial drug. So what was done for MMV 390048? They did the in vivo studies and they noticed that in the rat it was teratogenic. So they had hernia in the diaphragma and they had ventricular septum defect in these rats. They didn't see anything in the rabbit. So then you can imagine that the people start thinking, well, what is the impact for pregnant women? Is it a rat-specific effect or is it really a risk for these pregnant women? So as you could see from the third revision of the ICHS-5, one of the scenarios that you can apply is that you add on NAMs to increase your weight of evidence, so to see whether it's an effect or not. And that's what this company did. So they actually use zebrafish embryos and human iPSCs. And in those NAMs, the compound was actually not teratogenic. But, so these data were published, and you need to be very careful when you look here at this table. One of the things that I always tell my students is that you don't start with your evaluation of toxicity before you look at the exposure. So the exposure is the first thing that you bear in mind when you do your assessment. And what you can see in this table, so this is the rat. The compound was teratogenic in the EFD study at a 20 mg per kg doses. So that corresponded to a Cmax concentration of about 13. If you go below that, no teratogenic effect. If you go higher, of course, still teratogenic. The rabbit was negative, but if you look here at the concentrations in the plasma, then you see that you never reach the 13. So you are actually below that concentration. So that could be the reason why you have no effect in the rabbit, and they couldn't go higher because they had too high maternal toxicity. So the dams were suffering, so you couldn't increase the dose. What you then see is that the repro tracker, they could go to 15, so that's higher, so that's negative. So that's something that is opposing then the rat data. But again, for the zebrafish, only 8 micromolar could be used. So that's again below the teratogenic effect that you noticed. So I think you can see here that it becomes very complicated when you look at these data to make a strong case that this compound does not have an effect or would not have an effect in women. What could then be the way forward? And that's one of my last slides. So we actually embarked on a new venture. So here, I just want to show you, so this is a pregnant woman. So in most cases, we don't have any data. We may have data in pregnant rats and we may have data in pregnant rabbits. What we have already is human iPSC cells. So I showed you, for example, the repro tracker. What you could do, so of course you can compare those with each other, so the in vivo studies with each other. But let's say that we have already here the human NAM. So what you could do is compare the human NAM with the rat and the rabbit in vivo data. That's something that you definitely can. That's what's done. But if you have also a rat NAM and a rabbit NAM, then you can first compare them in between, so in vitro with in vivo. And then you can also compare all the in vitro assays with each other. So that brings me to this point. So we have already human iPSC cells. We already have rat ESC cells. They are commercially available. We have the rat in vivo data for several compounds and for rabbit data. So the missing puzzle piece is actually the rabbit ESC cells. And that's something that we are currently now developing in our lab, and it's actually part of an NC3R's grant in collaboration with Toxis and Astru Labs. So if we have all of the NAMs, so the human and the animal NAMs, then we can start comparing them. And let's say in the ideal scenario, that all of the NAMs are concordant, and they are concordant with the in vivo data, then we can be quite sure that the outcome in human in vivo will be very similar. Of course, this will not be the case for all of the compounds. So this brings me to my last 2 slides. So what we know now, if you think about NAMs for regulatory purposes in the field of developmental toxicity, it's only in certain circumstances that you can still do this. So it's still limited, but there are some openings. Also to bear in mind is that when you want to submit a NAM really for regulatory purposes, it should be qualified. The qualification is not mentioned into the ICHS 5, so how you have to do it. But we know now, of course, from EMA that there are now qualification criteria for several NAMs, and you can follow the same route to qualify your specific NAM. We also know that regulators are encouraging companies to include already their NAMs data when they submit the in vivo data. So even though they are not really used for the risk assessment or the safety assessment, by including them also, the regulators see the data and they can also learn from it. But there are actually two caveats. So at this point, there's no safe harbor. So companies are reluctant to submit them, especially, for example, if you have negative findings in your animal studies and then you have a positive signal in the NAM, companies are not really inclined to submit those data. And also it's on a voluntary basis, which means that there is a high risk for biased data that will be submitted, in the sense, again, that companies will probably submit the data where the NAMs are concordant with the animal in vivo data, and if they are disconcordant, then probably they will not submit those data. So this brings me to the general conclusions. So in most cases, we don't have clinical data in pregnant women for drugs and chemicals, so we need to rely on the non-clinical data. The data are still gathered in animal studies in most cases, but we see now that there are openings, so potentially in the future we will see that DevTox NAMs will replace or at least reduce the number of animals. If they are used in screening purposes, then this compound will also not be tested in animals if it's positive. And maybe the most important point that I want to make is that none of the individual NAMs is the Holy Grail. And we also should step back from looking at the animal studies as the gold standard, because by themselves, they are also not the Holy Grail to be looked at. And again, so the NAMs, when you want to use them, they need to be qualified, and there needs to be a very clear context of use to be provided. And here you have some more information. So our essay on the database, my contact details, and there's actually now a workshop on DevTox NAMs in Washington at the end of September, 1st of October, and there regulators, people from industry and people from CROs, academia will all be there, and we will try to move this forward in the field of DevTox. And that brings me to my last slide of this presentation.
Thank you very much, Steven, for this very nice and extensive presentation. So far, I don't see any questions in the chat, so don't be shy. You can put your questions there. But I already actually had one question, and you more or less answered it already, because in the beginning of your presentation, you indicated that from 2000, there were already several NAMs validated by ECFAM, but they are not used. So what do you think is the reason? Because if we talk to regulators, then we often hear the comment that the reason why NAMs are not being used is because they are not validated. But if they are validated, what do you think is then a major threshold for them to start using it?
Yeah, so the validation effort by ECFAM was done just by 20 chemicals. And for those 20 chemicals, the data were conclusive. A few days later, companies also tested pharmaceuticals, and there it appeared that, so it was mainly the mouse embryonic stem cell test, so the limpet micromass assay has actually not been really used. And also the rat WAC has then been tested with pharmaceuticals, and the outcome was not really good. So the concordance, and that's one of the reasons why they never actually progressed. So people stopped it. And then, of course, I mean, if you only look at cardiomyocyte differentiation, that's just one of the germ layers that you assess. So if you extend it, so that's, I didn't explain it in my talk now because of time limits, but actually in our essay, we will look at ectodermal, mesodermal, and endodermal differentiation. So the three that you need for organogenesis. So that could also be the reason why the data were less conclusive. So that's why the assay should be really qualified, and the context of use is there very important. So I think we need to step away from an assay that will be able to cover everything. So if you have an assay, for example, which is performed for a certain class of compounds, then this is already a step forward. So then this type of compounds will be able to assessed by the NAM, but it will be impossible to cover all therapeutic areas and all chemical classes with just one NAM. So that's I think what people are now starting realizing, that this will not be possible, and that would of course be the holy grail, because that's the easiest way, but that will never be happening. And then, not to complicate it, we are also still missing the fetal maternal interaction with these current NAMs. So potentially we need, like you have now, a multi-organ on a chip setup. Potentially we will need like a placental component added to the current NAMs, where you can then also see whether there's any interference from the placenta on the outcome.
Yeah. And because I also like that you immediately indicated, like, what can we do now that we have, and from a regulator point of view, you need to have a safe harbor. So what would you think would be a low-hanging fruit to stimulate regulators and industry to start using them? You come from industry, so
it's a difficult one. I know that the developers, of course, are very eager to do this. So there are now, I think, informal discussions between developers and the European Medicines Agency, because that's possible. You can already have talks before you do your real qualification to see what is actually needed, what are the pitfalls and these things. So that's actually ongoing. To be honest, I mean, we actually don't really know what is really preventing companies to do that. And that's actually one of the reasons why we have the workshop in Washington, where all the companies will be there to really get a grip on, why are you not doing this? Why are you not even submitting your NAMS data that you have from screening alongside your in vivo data? Because that's a possibility. If you have the safe harbor, that should be definitely something which is possible. So it's a very good question, but we don't really have the answer to that one yet.
Yeah, I'm really, because you also indicated that for in the beginning of your presentation, there was a lack of standardized protocols, but then at the end with the staining, you saw that it was very difficult. Like, do you see also like low-hanging fruit to have a standardized protocols, like, because there was a lot of research going on the zebrafish embryo assay, like maybe for the metabolic assay? Do you see there like an opportunity that the research group starts working together to reach this goal, or?
I think from an academic point of view, that should definitely be possible. And I think just by also sharing that information, like we did, just completely sharing our protocol so that people can have a look at it, that's possible. But within companies, it's much more difficult to change. And sometimes it's really very small things. So it can be something small in the medium. And then because they already evaluated their compounds, their proprietary compounds with that protocol, that was actually one of the goals of our consortium that we had when I showed you the paper on the biological variability. We actually started off with having an assay which would be the same in all of the labs. We didn't get to that point because the companies are not willing to do so, because they have already tested their compounds with their protocol.
Yeah. Okay. Thank you very much, Steven, for the nice presentation. I'm just taking a look at the chat, and besides a lot of requests for the certificate, I don't directly see any burning questions for you. Okay, so in case somebody still has questions, don't hesitate to let us know, and I can still forward them to Steven after the webinar. So again, thank you, Steven, for your very nice and clear, interesting presentation. So now it's time for the presentation of Dr. Diana Delbany. So Diana obtained her master's degree in cancer biology at the University of Beirut, and she completed her PhD in biomedical and pharmaceutical sciences under Professor Mark Parmentier in the Institute of Interdisciplinary Research in Human and Molecular Biology at the ULB. And since 2021, she joined the research team of Professor Claudia Spitz at the VUB, more in particular in the group of genetics, reproduction and development, where she works as a postdoc, mainly on brain organoids models to study the tumorigenic capacity of human pluripotent stem cells. Diana, the floor is yours.
Yes, hello. Thank you, Mika, for the introduction. I'm super happy to be here today. I will share my presentation. And if you can please confirm that you can see it in presentation mode.
Yes, we can see it.
Okay, so thanks again for the introduction. Hello, everybody. Indeed, my presentation today will be entitled Brain Organoid as a Versatile Model to Study Tumorigenesis. So I'm going to start with a general introduction. So Voila. As you all know, our human brain is a very complex organ, yet it's a very fascinating structure. The human brain consists approximately of 87 billion neurons. Each of those neurons should be generated at a specific time, also should be migrating into the correct location and form a precise connection in order to form a normal function. Despite this complexity, the brain or the human brain originates from a very limited pool of stem and progenitor cells. And even those cells, they need to be developing according to a very specific and predefined rules. Interestingly, the pace of brain development vary a lot across species, and for the human, this is a very prolonged process, as we can see this figure here. So it started during the embryonic period and then continued to the fetal period, and it's also prolonged until adulthood. So our human brain take much longer to grow, to mature, and also refine their connection, which is approximately 30 years, representative of almost half of our average lifespan. In general, most of our understanding for the human brain development largely come from experiment and studies that has been done in rodent model and particularly in mice. So when we look at the mouse neocortex, it forms actually from polarized neural epithelium, and also the cells are organized from apical to basal axis. I don't know if you can see my pointer. So from basal to apical side. So at the basal side, we have the cortical plate where we have the neurons and their axons, and also at the ventricle side, we have the progenitor and the stem cells. Also, we know from mouse model that those cells can divide symmetrically, so they can enlarge the pool of progenitor cells, and this symmetrical division happen early in development, and also they can divide asymmetrically to give rise to progenitors and also to neurons, and this is when the neurogenesis start. When we compare this to a human, actually, we have some differences. So in the human brain, we have an extra layer, which is, we call it the outer subventricular zone, which is here. It's largely absent in rodent. In this layer, we have the outer radial glial cells. Those cells act as transit amplifiers, and they divide asymmetrically to give rise actually to progenitors or intermediate progenitors and to many neurons. This is why our human brain has more or high number of neurons when compared to rodent, and in general we have a larger cortex. So these differences in general limit the use of rodent to model human brain development, and also we know that some neurodevelopmental disease cannot be modeled in mouse, and eventually, of course, the excessive use of mouse for experiment and studies have a lot of ethical concern. This is why, indeed, we have a critical need for advanced human-based model. So when we look at our option, the first option we can think about actually is the use of a human fetal brain tissue. This can be the most representative or give the most direct insight, but of course we know that this has a lot of ethical concern and it's very hard to obtain. The second model that we can think about is the use of human pluripotent stem cells that can be differentiated in 2D culture. This approach can be useful actually for patient-specific phenotype, but again, the cells, when they are cultured in 2D, this lack the 3D structure of brain, which does not mimic the in vitro brain development. So this leads us to the third option, and actually this is the most advanced technology, the use of organoid that can recapitulate physiological features and also functionalities. And most importantly, they are a valuable tool to study the development and also to remodel some tissues like the brain and the retina, and those tissues in general, they are having a very difficult cell retrieval for further analysis. Of course, the brain, for this talk, I'm going to focus on the brain organoid. So the brain organoid, we start from a human pluripotent stem cell culture that we aggregate into embryoid body, and then we induce to neural differentiation. This induction can be guided or unguided. There's several protocol and let's say several schools for the kind of differentiation. And then eventually the organoid can recapitulate the human brain development and recapitulate specific feature such as the cellular diversity and the layered organization. In parallel for those studies using the organoid as a model which overcome the limitation of mouse model, especially for the neurodevelopmental disease, we have also increased clinical trials specifically for the brain diseases where we have the use of stem cells to treat some condition, for instance, sclerosis, multiple sclerosis, spinal cord injury, Parkinson's disease, stroke, and others. There's plenty of clinical trials with a very promising result. So why I put these two strategies together in one slide? Because there's something common, and the common thing about these two technology is the use of human pluripotent stem cells. So when it comes to those cells, we all know that those cells are very powerful cells. They are pluripotent. They can differentiate to any cell type of the human body, and they also have self-renewal capacities. But something people maybe don't know or ignore sometimes, that those cells during their expansion and their culture in vitro, they can acquire chromosomal abnormality or aneuploidy. So after for instance, for the embryonic stem cells, after their derivation or during the reprogramming of iPSCs and the expansion in vitro, those cells can acquire chromosomal abnormality. Those then we can consider them as more or less mutant cells. They will stay in the culture. They will also be present when the cells are differentiated, and also might have consequences either when it comes to transplantation in patient or when it comes to modeling using organoid model. So what do we know about those aneuploidy? Some of those aneuploidies are well characterized, but other are not. We know that they can provide excessive proliferation potential, and also they might evade apoptosis. So here I will share a map of the most frequent chromosomal abnormality that we find in both embryonic and induced stem cell culture, where we can find the gain of chromosome 8, we can find the loss of chromosome 18, the most frequent, which is the gain of chromosome 20, 12p, and 1q. So those chromosomal abnormalities are very frequent, and they have functional consequences. Not all of them are very well characterized. So in our lab, we do actually characterize those chromosomal abnormality, and we want to understand what are the impact on the proliferation, on the differentiation, and so on. So why we want to be aware of these chromosomal abnormality? Because, for instance, this is a recent study that has been published, and where they show that the use of iPSCs derived beta cells for diabetes treatment eventually led to the formation of a mass at the site of the injection. This mass was, the patient also have enlarged lymph node. This mass was having a rapid growth, also metastasis to the lymph node. This mouse did not respond to chemotherapy treatments, and the only solution was the surgery to remove this mass. And as you can see how big this is, so this is considered as a teratoma. The analysis of this mass showed that there's plenty of mutation. They didn't show that this tumor had some chromosomal abnormality, but what if indeed those cells carried chromosomal abnormality? Would the patient had a worse phenotype? Would it lead to a worse case scenario? This is something we wanted to investigate. Also, people, the aneuploidy is a hallmark of tumor cells. So this is the karyotype of a normal diploid cells where we have 2 copies of each chromosome. But when we look, for instance, at the karyotype of a glioblastoma, we can see that we have a quite chaotic karyotype. We have several copies of each chromosome. So in general, the mutant pluripotent stem cells or pluripotent stem cells that carry chromosomal abnormality, they are characterized by this genetic instability. They have increased proliferation, and they have also decreased sensitivity to apoptosis, which make them closer to cancer cells. But up to now, there's no proof that indeed aneuploidy can prime tumorigenesis in the stem cells or in the differentiated stem cells. So this is a hypothesis that I wanted to investigate in my project. Does this aneuploidy prime the transformation of stem cells differentiated in brain organoid? Yes or no? And we chose brain organoid because there is plenty of clinical trial using the stem cells to treat neural diseases or neurodevelopmental diseases. So to investigate this hypothesis, actually we used, we were based on a publication that was published in 2018 by Bayen and his colleague. In this paper, actually, they proposed a protocol where we start with the stem cell culture and then eventually the aggregation into embryonic body, neural induction. And at the end of the neural induction stage, they induce mutagenesis, which means that either they overexpress an oncogene, or they downregulate a tumor suppressor gene, or they make a combination of mutagenesis or genetic modification, and then they observe whether those organoids will form tumors, yes or no. So in this paper, as I was saying, so they use different kind of mutagenesis cocktail. So for instance, here I will emphasize on the overexpression of CMYC. And indeed, this mutagenesis or those genetic mutation that they induce on the brain organoid induce the formation of tumors. And also, interestingly, depending on the type of genetic modification that they induced, the organoid can have distinct transcriptional profile and cellular identity. And those actually results highlight how valuable this tool is and how can we actually change the condition to obtain what we want. If we want to study glioblastoma, if we want to study a central nervous system neuroectodermal tumors or others. So it just depends to tune actually the mutagenesis cocktail that we are using. And also they finally proved that it's not only some cells that they are just green and they are proliferating in the organoid. So they indeed also confirmed their tumor initiating capacity by engrafting those cells in mice, where those cells indeed they were able to engraft, they were able to survive the procedure and to expand in vivo, and also importantly to maintain their subtype identity upon a transplantation into mice. So to get back to our hypothesis, so I applied actually this technology or this protocol. Unfortunately, I cannot share a lot of results for today because this is work under like the paper is under construction. So I am obliged to share limited amount of results, but indeed our results showed that we have higher transformation rate when we form organoid with aneuploid cells or with mutant cells compared to brain organoid formed with control embryonic stem cells. What I mean by control, it's those cells that they are genetically wild type or balanced. They don't have any kind of mutation. So to conclude, as a take-home message, so brain organoid model combined with targeted mutagenesis can be used as a powerful platform to test tumorigenic capacity of different gene aberration. Also, although the field of stem cell-based therapy continue to advance and we have several clinical trials, it remains essential to implement more rigorous and sensitive screening to ensure the safety and the good quality of the human pluripotent stem cell culture and the product. And it's also very crucial to minimize the risk of transplanting genetically abnormal pluripotent derived cells, as we know from our current work that they might risk forming a cancer or they have a tumorigenic potential. Thank you for your attention.
Thank you very much, Diana, for your interesting presentation. So far, I don't see any questions in the chat, but I was wondering, what are the next steps in your research that you envision?
Yes, so now this paper is actually, there's two papers in the preparation for this project. Now that we know that aneuploidy actually have effect on the tumorigenic potential and also can have an impact on the differentiation of those cells in brain organoid. But we want, or I want actually to benefit from this system. For instance, to study a different type of brain tumors, because maybe depending on the chromosomal abnormality that we have, we might have different phenotype of tumors, and that maybe recapitulate the diversity and the heterogeneity that we have of brain tumors, which is not always very easy to be represented in vitro as it is represented in patient. So maybe those chromosomal abnormality will help us to have more insight on this aspect.
Yeah, and I see we also had a question already from Mark. He asked if you have already checked the metabolism capacity of your model.
No, no. Actually, with this model, there's plenty of things we want. We can check. Indeed, metabolism, one of them. So actually, now we for the papers, we are conducting big sequencing experiments. So big, what I mean by big is that we are investigating several lines of stem cells and also all the mutation. So we want to see what can they be, how can they be different if they are forming different type of tumors. And also something I didn't mention, that we see also that aneuploidy can affect their ability to differentiate. So if they can affect their ability to differentiate, it might also affect their other ability, proliferation and things like this. But specifically metabolism we didn't do, but we might actually end up during the analysis on something also interesting that might be like new avenues for new project.
Okay, that's very good to hear. I don't see any other questions in the chat. So again, thank you very much, Diana and Steven, for your nice and interesting presentation. Also, a big thank you to the attendees for coming to the webinar and following it. So we will provide you the certificate of attendance within 2 or 3 weeks. If you still have any questions for Steven or Diana, don't hesitate to send them to us. We will then further distribute them. When you leave the webinar, you will also receive a link to a short survey where you can give feedback on the speakers and the topics of the webinar. So don't be shy to provide your comments. You can also indicate if you would like to have a particular topic in the future, so for our next series in the fall, or on the other hand, if you also would like to present your own work, don't hesitate to put it in the comments. And if you want to be kept updated about our future activities, don't hesitate to register to our newsletter via our website. So again, thank you very much to the speakers and attendees for coming to our webinar, and have a nice evening. Bye.
Thank you, you too. Bye.